I'm Lan Zhao, a postdoc from Stanford University. I'm a bioinformatician working on microbiome-related projects. Today I'm very glad to present our research works on the rat-microbial BodyMap across 11 tissue types and 4 developmental stages. This is a rat transcriptome dataset we used in this study. A total of 660 samples were collected across 11 rat organs and 4 developmental stages. The original authors had already investigated the rat transcriptomic landscapes based on the mapped race. We expanded their research on the unmapped race as well, in order to identify rat-related species in different tissues. And in the same time, we are interested to identify age-related species. This is a pipeline we used for RNA-Seq based microbial data analysis. The input data are the FASTQ files. After mapping those data to rat genome, we found that a small percentage of the race were unmapped. After quality control, those data were considered to be the microbial data. And subsequently, we used the Kraken tool for microbial taxonomic classification, which can help us generate an OTU table, which is the operational taxonomic unit table. We also performed a decontamination pipeline in order to remove potentially contaminated species in the OTU table. Our following analyses include clustering and also the downstream visualizations. A total of 4,647 species were identified based on the 1% prevalence threshold. In this table, we listed the top 12 phylum identified in these 11 tissue types, which is ranked according to their population size. And this bar plot shows the relative abundance of the 12 phylum in these tissue types. We can see the firmicutes and the proteobacteria are the two most abundant and diverse species identified in the rat tissues. Next, we performed a beta diversity analysis in the 4 developmental stages. From this 3-dimensional field principal coordinates analysis, PCOA plots, we can see the lung samples showed notable separation from other tissues in the juvenile, adolescent, and adult stages, but the separation was not visible in the elderly lungs. We also performed unsupervised classification in order to identify rat-microbiome interaction patterns. In this microbial abundance heat map, each row represents a species, and each column represents a sample. You can see 4 distinct and well-separated rat-microbiome interaction patterns, both in this heat map and this PCOA plot. The table below the heat map is a contingency table for tissue distribution of the 4 patterns. We can see the liver and muscle samples were basically classified into the P2 pattern. Lung samples were classified into the P1 and P3 patterns. No significant enrichment can be seen for developmental stages distribution of the 4 patterns. This bar plot shows the taxa abundance in the 4 patterns, which agrees well with our previous results. So thank you so much for your attention.